Hello, I'm Dr. John Kruse, and today I'm going to be talking about the gut microbiome and its possible contributions to ADHD. So I'll be talking for about 20 minutes or less. If you have questions, you can type them in. This will also be posted on YouTube and Facebook, and you can write in questions later. So part of this talk was prompted by a study that's pre-released, so it's going to be released, but so the take-home message was from the study. Sorry for the background noise. The take-home message was that the microbiome at 6 months old in a group of 300 kids predicted which kids at the age of 10 years old had ADHD. So the question is this a really interesting, meaningful finding, or what does it mean, and how important is the microbiome to ADHD and other aspects of mental health? So to back up a little bit, for those of you who haven't been tapped into the zeitgeist for the last few years, what is the microbiome, or what the gut microbiome? So the gut microbiome is the collection of non-human cells, living organisms, mostly bacterium, but also archaea, viruses, fungi that exist in our gastrointestinal system. And our whole microbiome is all those forms of life that are all over our body. So our skin hosts some, other parts of our body, they're there, but the biggest concentration is certainly in our gastrointestinal system, and far and away mostly in the colon. The acidic environment of the gut and the small intestine precludes heavy growth there. So the size of the microbiome in our gut, in our colon, is about 200 grams. So it's only about a sixth the size of our human brains, which are about 1,300 plus grams. But it entails more than 100 trillion cells. So that's more than 1,000-fold more cells than neurons exist in our brain, or brain cells exist in our brain. So it's an incredible number of cells, a few hundred different species of particularly bacteria, and bacteria has been the group that's most studied and are certainly the major component, but that's not to say viruses and fungi and other archaea may be also terribly important. So a lot of different cells. So even though there's a big diversity of cells, about 90+% of the individual cells are only from 30 to 40 different strains of bacteria. So there's a lot of diversity, but it isn't that of those several hundred to a few thousand of variety, they're each having a sizable share in the total. So a few other things about this microbiome, I sort of contrasted it or compared it to the brain. In the brain, most of our neurons that are present in young adult life are the same neurons throughout life, whereas the microbiome, there's tremendous turnover. Actually, about almost 2/3 of your stool is dead bacteria. So there's a lot of turnover. We know there's circadian fluctuations in which bacteria numbers are more present at certain times of the day. We know there's big developmental changes. And the source of the bacteria and other parts of the microbiome are environmental. So these are living organisms, are not spontaneously generated. They get there somehow. Diet seems to have a big role in what your microbiome is, your local environment in terms of where you're living. So they do dust samples and assay what bacteria, what fungi are there. Those are strongly correlated with what is present in bodies living in those states, conditions. We know that whether you're born through cesarean section or vaginal delivery greatly affects your microbiome, and that in general the vaginal delivery results in a much broader, richer range of bacteria growing there. And we also know that drugs during your life, particularly antibiotics, have been strongly looked at, shape what your microbiome is, but certainly other medications that are taken on a chronic basis can affect the microbiome, and certainly other toxic chemical exposures can affect what's growing in your digestive system. So a lot of the interest in the last few years on the microbiome is how the microbiome affects and is affected by the central nervous system. So this is a two-way system. This isn't just one thing affecting the other, and again, it's a dynamic, changing system. But clearly, during early development, the microbiome has an influence on how your hypothalamic pituitary adrenal system develops. It has an effect on how the nervous system itself develops. So a big source of what's called brain-derived neurotrophic factor is actually microbiome bacteria produce chemicals that affect brain cell growth. And the microbiome also affects the immune system and its development. And in particular, having a healthy microbiome is important for developing both what's called the gut-body barrier. So earlier in life, more of what we are exposed to through our digestive system winds up in our bloodstream within the first 2 years of life. Not only is the cell-cell connections in the lining, the mucosa of the intestinal system developed to exclude many larger, potentially toxic chemicals, but also the microbiome itself is part of the barrier preventing just anything that you ingest from winding up automatically into your blood system. And also the development in the brain of the blood-brain barrier is dependent or connected with a healthy gut biome. So some of the ways, so that's what's going on during development. During ongoing, adult life, what does the microbiome do? I mean, why do we have so much sitting around? It must be presumably doing something beneficial for us. And clearly there are major roles for both absorption of nutrients and digestion, metabolism of nutrients, breaking down of certain foods, substances our own cells in our own body can't do. We're used to thinking of cows being able to eat grass or termites being able to eat wood. And again, in those species, it is their microbiome that allows to break it down and eat things that we don't think are edible or amazing or, normal food items. But again, many of our own foods, it isn't our own cells in our own body that are breaking down or digesting. It's our microbiome that is breaking them down and digesting and metabolizing them for us. And there's been a lot of research on different microbiomes and whether and how they predispose certain people to obesity or other conditions because, again, specifically how they're breaking down and absorbing nutrients. Another ongoing feature of what the microbiome is doing is that some of the chemicals they're producing are precursors to the neurotransmitters, and particularly producing tryptophan, which is a precursor of serotonin, and producing phenylalanine and tyrosine. Phenylalanine also is converted by both microbiome and our own cells into tyrosine. Tyrosine then goes to be produce both dopamine and norepinephrine. So the three neurochemicals that we talk most about, serotonin, norepinephrine, and dopamine, are all large amounts of their precursors are made in our microbiome. And in terms of serotonin, more than 90% of the serotonin in our body is being made in our digestive system through our microbiome. Serotonin was actually first discovered in guinea pig small intestine. So the thought is that the production of these precursors of neurotransmitters may have important functions in terms of how smoothly our brain systems are working, given that these are important neurochemicals. And again, this is not just a microbiome producing things that affect the brain. The gut has an extensive neurologic system, the enteric nervous system, which is the second biggest collection of nerves outside of our brain. So many of these, the vagus nerve coming down from the brain, or it goes both ways, from connecting the brain to the digestive system, carries information both ways. So taking information from the digestive system up to the brain and sending information from the brain down to the digestive system. So a lot more research on the microbiome has been done with autism than on ADHD. There have been a few studies looking at ADHD and seeing if the microbiome is different in people with ADHD than with people without it, and many of the studies have found some differences, and there haven't been complete consistency among the differences. But the study I'm going to go into more detail in a second is a study done by Andrea Cassidy Bushrow and her colleagues at Henry Ford Medical Center in Detroit. And what they did was look at a group of more than 300 kids. They took samples at 1 month of life and stool samples at 1 month of life and 6 months of life, analyzed the bacterial composition there, and not just bacterial, but other organisms, including fungi, and then looked at age 10, which kids had ADHD and which kids didn't. And what they found was that there was a correlation with the greater diversity at 6 months of age of bacteria actually correlated with ADHD. You would think that greater diversity is often taken as a sign of greater health, and why that would be correlated with ADHD itself rather than not ADHD. Their assumption is that that was a marker of the gut being prematurely developing. And there was also some association at the sample size or sample diversity, sample groups of bacteria and fungal. There are 18 types of bacteria and 3 types of fungi present in amounts at 1 month of age that seem to correlate or be associated with ADHD at a later time point. And by 6 months of age, they identified 51 different types of bacteria and 3 of fungi that were correlated again with developing ADHD at a later, by 10 years of age. Now, one of the most simplistic explanations for all this, we know again that there's a high genetic component to ADHD. A simplistic factor would be these genetic factors are both controlling how your microbiome develops and what your nervous system is developing, and that these may just be different manifestations of a genetic system. So again, this study in itself doesn't show that these microbiome differences caused ADHD. That's certainly plausible. One thing, given the time course, that this doesn't allow. So we have an association. Again, association or correlation isn't necessarily causation. What we can say here, though, is that ADHD isn't causing these differences in biome, given that the timeline is different, that this was ADHD at 10 years of age. That didn't go backwards in time and create different biomes early in life. So other potential issues and issues with the whole field, I mean, one is, as we mentioned, so most of the time we are assessing the microbiome rather crudely, and crude at a couple levels of the word. So one is we're taking stool samples, and anyone who's had parasitic diseases or had extensive gastrointestinal workup because they've had GI problems or diseases or issues may be familiar with, if you take a blood sample, blood is a constantly churning, evenly mixing system. So if you're looking for COVID viruses, if you're looking for immune responses, you can take a sample of blood. It's pretty well representative of blood you would find anywhere within any veins, arteries, or capillaries. I mean, the oxygenation difference will be there between arteries and veins. But in terms of substances floating around, it's a well-mixed system. Stool, on the other hand, is not a well-mixed system. It's not it's a tube that's pushing things out. So if we're relying on a stool, that's why when you're searching for parasites, sometimes the first few stool samples or the sample they take within the sample of stool itself, they don't find anything. Stool, again, there's some mixing, but it is not a wonderfully universally, mixing things around like the dryer in your washer-dryer system or the washing machine itself. So that brings into question sort of how representative the samples are. Two is presumably there are different, well, not presumably, we know there are different environments during the course of the length of your colon. So there's differences in acidity, there's differences in water content. Presumably some of those environments are better for some types of bacteria than for others, but a stool sample is collected at the very end. So again, how accurately it's collecting or assessing what's going on in your whole microbiome is open to question. So just a little bit more technologically, so what we're doing, the common way of assessing bacterial content of those stool samples is by using chemical assays that are looking at the 16S ribosomal RNA, and that's a highly conserved evolutionarily it's highly conserved, so we can identify what types of bacteria there are by what types of 16S ribosomal RNA we're picking up. But it's not necessarily there may be differences we're missing, or there may be differences we're confusing or blurring. There may, in the long run, turn out to be better systems for assessing the diversity of bacterial life in your microbiome than what we're doing currently. A few other things, so there have been a few other studies of microbiome diversity in ADHD. There is not general consensus in them. There's also a small number of studies looking at can we change the microbiome by usually, I'm blanking on the term now, usually different forms of yogurt, different diets, different probiotics, different probiotics. And my oversimplification from the probiotic field is that certainly if you are introducing different sets of bacteria, changing acidity, doing other things, you absolutely can change your microbiome. Most of the studies suggest that once you stop that intervention, there is a strong tendency for an individual's microbiome to revert back to the status it was before that dietary intervention. There may be more profound or effective ways, and this is not particularly in brain conditions that have been looked at, but for some other autoimmune and immune gastrointestinal conditions that have been looked at, actually doing stool transfers, so giving capsules that are sealed. They aren't opened up until they're far along in the digestive system. So fecal transfers may be more, maybe a way to give a more sustained change in someone's microbiome. Again, certainly if someone takes on a sustained dietary change, or again, even a change in your environment, because again, we know we are taking in bacteria, fungal, viruses, other things from our environment, and if we're in a different location, that can have an impact. So we are still clearly at the early stages of studying the microbiome, and particularly studying the interaction between the microbiome and the brain. It certainly is going to be, and already is, an important factor in development, and given the ADHD and a host of our other what we call psychiatric conditions or diseases are developmental in nature, and at least have, even if many of them, all of them have at least some genetic contributions, some of them very strong ones, there are clearly other environmental factors that are at play in whether a given individual is going to display these conditions. So we have much yet to learn, although again there are already some tantalizing early results. I'm going to take a break in August as I move to Hawaii, so I'll be returning in September, and stay tuned. That's all. I'm not seeing any questions popping up, so bye for now.